All right, welcome to the Lab 5 overview. I'm going to start with the PDF file. You can see it begins with a quick introduction into Protus. So this lab focuses on the kingdom Protista, but as it mentions in this introduction, that kingdom is being removed along with the other three kingdoms that fall under the domain eukarya, which we'll visit in chapter 28 in lecture. Activity 1 focuses on defining terms. There's some help in the PowerPoint with this. You may need your lecture notes and also the internet, but I'll show you in a second. There is a lot of information in the PowerPoints to help you out with this. Then the second activity, you're going to be focusing on the four supergroups, okay? And I have these in the PowerPoints, but the first supergroup you're going to focus on is your excavata. And the examples I give you are euglena and trypanosoma gambians. There are two figures for euglena, okay? Make sure that on both figures you can identify these three structures that are found on euglena and label those. For trypanosoma gambians, make sure you can locate where the organism is as well as its flagellum. I don't have it listed here, but also point out the red blood cells just to differentiate. The second supergroup, as we'll discuss in lecture, will be the SARS clade. The S stands for straminopiles. So your examples of straminopiles are your marine diatoms and your sargassum or brown algae. From the marine diatoms, it's this figure down here. Okay, what I want you to figure out is whether every specimen in this figure is a diatom or whether only certain ones are. If you believe all of them are diatoms, then just write all diatoms across the top. If you believe only certain ones represent diatoms, okay, then draw little arrows pointing to them to let me know. Okay, so that's up to you to figure out. And then for sargassum, there are four structures you need to identify in sargassum. So you're going to need both of these figures here, okay, to locate all four. So you won't label all four of these structures on both of these photos. Combined together, you should be able to find all four. Okay. Now the S stands for straminopiles. The A stands for alveolites. The alveolates consist of our dinoflagellates, our paramecium, our plasmodium falciparum, which causes malaria, and stentor. Okay. So for your dinoflagellates, that'll be this figure here. I want you to treat it the same way you do the diatoms. Okay. If you believe every specimen in this figure is a dinoflagellate, then just put all dinoflagellates. If you believe only certain ones are, draw little arrows pointing to them to let me know. Now for the remaining four figures, it'll be up to you to figure out, okay, which one or ones are paramecium, plasmodium falciparum, and stentor. Okay, so make sure you put the names of the organisms next to them and then make sure you identify the specific structures. Okay, and if you notice here for plasmodium falciparum, it does indicate, okay, make sure you label the red blood cells and make sure you differentiate between the ones that are infected and uninfected. Well, the ones that are infected, that's where you want to point out where plasmodium falciparum is, okay? And then stentor will be able to identify these two structures. So just label them on these figures here. Now the R in SARS clade stands for rhizarians, but I didn't give you any samples by mistake or just by chance. I overlooked them. So I moved on to the last two supergroups. Archaeplastida, which consists of our green algae, such as our chlorophytes volvox, which is a figure here. Simply, volvox is considered multicellular in nature. I give you also in the PowerPoint, not only do I give you additional information on these organisms, these protists, but I also give you some web links. A couple of them take you to videos, okay, to give you videos of these, and one of them is a real good one on Volvox. I think it is. It's pretty simple. It'll help you label the two different types of cells, okay, or colonies that form Volvox here. So make sure you label those. And then for the last supergroup, it's called Uniconta. As we'll talk about in chapter 28, this last supergroup, Uniconta, not only consists of amoeba-like organisms, but it also consists of our animals and our fungi. So that's where the kingdoms are being reclassified. But for now, this is an amoeba called Chaos. So I give you a video on this too if you want to take a look at it, which will help you label the two structures on the amoeba you need to. Now there are four additional questions you need to answer. Okay, I tried to provide some additional information in the PowerPoint to help you with these questions. But again, if you don't feel you find them there, use the lecture, chapter 28, okay, to support you with this lab answering questions and labeling, as well as use the internet for these. Let me see if I can, trying to bring up the PowerPoint right now. Okay. So I'm not going to go through the PowerPoint step by step, but I do have the PowerPoint there. It gives you the introduction. What is a protist? A eukaryotic organism that is not a plant, fungus, or animal. Living protists, along with plants, animals, and fungi, represent descendants of an enormous adaptive radiation that occurred after the evolution of the eukaryotic cell. And this is a part where it talks about that we'll focus more in chapter 28 in lecture. Protista is falling from use as a kingdom name. Nonetheless, it's a useful term for a very large group of organisms. So there are going to be some terms you need to identify define in activity 1 of the lab. Polyphyletic is one of them. A couple of other terms you're going to have to define are monophyletic and paraphyletic. So I give you some information here, but I also give you a web link to help you define those terms in activity 1. Okay. These are the 4 supergroups. You'll see this a whole lot when we get to chapter 28. Okay, some of the organisms you can see directly here, such as diatoms and dinoflagellates, just to let you know where we're going here. And then I give you some information on each of the organisms that you have to ID. So you should have all the information you need to help you answer and label, okay, correctly on these, okay? If you're going to make a mistake, make it on the side of you put too many labels and not enough labels, okay? When you give an answer to a question, make sure, again, if you're going to make a mistake, make sure that you gave a little too much information, but preferably you study the information so you know exactly what to write to answer those questions at the end especially. And then I give you some additional information on some of these, such as Plasmodium life cycle and things of that nature. Here's a YouTube link for Volvox. Watch this. It's a very interesting link. It's a short video. It doesn't take very long. I think it's less than 2 minutes. I give you another link for Amoeba, which is also a YouTube video. It's a very short one. Watch this video. It'll also help you label your figure as well as that. It's simple labeling figure. I said this, I believe, in the last lab overview. Normally in person, okay, it's a lot more tedious, a lot more detailed, and we do use the whole lab time, and it drives a lot of the students crazy. But unfortunately, this is one of those labs, it's hard to translate to online. So I'm still working on it along with the other professors. Some professors, they're going above and beyond, but I haven't done that yet right now. It's been enough of a challenge for me and especially some of my students, okay? All right, that's all for the Lab 5 overview. I'm going to stop the recording. Y'all take care. Stay safe. Bye.